Identify a research problem. First, how to select a topic for your research. What is meant by research? What is the purpose of including this research project work in your M.Pharm course? These things let us discuss one by one. Next slide, please. So, role of research in postgraduate pharmacy education. Why a research component has been included in your M.Pharm program, right? There are main three objectives for that. First one is students, postgraduate students should be different from undergraduate students. Postgraduate students are supposed to solve the complex problems which are presented to you in the working environment where you are going to be employed in the coming days, right? So first, to develop scientific temperament. Scientific temperament means how to carry out a particular study by following scientific protocols, by following established procedures, methods. So if you know all these things, then it will become very easier for you to draft all these particular things. Just wait a second. So bridging theory and practice, which means whatever that you learn in the theory course for 2 semesters, lot of theory you might be learning, and how to put this theory into practice. Suppose in pharmaceutics you might have studied about novel drug delivery systems. Novel drug delivery systems, how exactly we can prepare novel drug delivery systems like transdermal patches, buccal patches, or solid lipid nanoparticles. If you are studying all these things in practice, in reality, in lab, how you will be able to apply whatever the theoretical concepts that you have studied in your project and come out with a good outcomes, right? So next one is publication oriented research mindset. Publication oriented research mindset in the sense, so whatever the topic that you have undertaken for the research, whether it can have publication potential or not. Generally, research publication is accepted only if your topic is novel, only when you contribute something differently to the existing knowledge, then only the eminent and one of the better journals, they will accept only when there is a quality in your work, otherwise they don't accept, right? So publication oriented mindset you have to develop. For that only this particular seminar has been designed. Next slide, please. So what is research? So first, basically you should understand what is meant by a research. Research is what? Research is not just searching again. Research means searching again. It's not searching again. Research is systematic. Research is scientific. Research is structured. So you have to systematically investigate a particular problem, right? First, define the hypothesis. Hypothesis means what you would like to research, what you would like to carry out, what is the problem which you would like to undertake. There is a difference between hypothesis and theory. You might have studied about theories, hypothesis. Hypothesis means assumption. Suppose to develop one particular research project, we assume that so and so compound may act on so and so disease. This is our assumption. In this particular assumption, what you do? You design one structure and you synthesize it, and you feel that depending upon the previous data, depending upon the literature survey, you scientifically guess that predict that this particular compound may show its efficacy on some of the diseases or some of the whatever it may be, right? So that is hypothesis. And once you carry out your studies, once you have statistically significant results, then your hypothesis is converted into a particular established concept, right? Otherwise it remains as a particular assumption only. Then the assumption becomes valid only through statistically significant results. That's why statistics also play a significant role in the research project, right? So next, it generates new knowledge in the sense whatever that is not known. So to discover something, research is to discover something, to establish some facts, to verify some facts, to add some new knowledge. This is the purpose why we carry out the research. First thing is, research means research is a systematic investigation. Systematic investigation in the sense, whatever the protocol procedure that you develop should be scientifically valid. It should have some scientific base, rationality. Otherwise, that is not at all called as a, you should proceed in a systematic manner, scientific manner, in a structured way. Otherwise, it cannot be called as a research, and in that research, you should be in a position to develop one hypothesis. Hypothesis means that assumption you have to develop, right? And to prove this assumption, you should carry out some experimentation, and through experimentation, you will get some data, and that data, if it is statistically significant, then only whatever the problem that you have identified, whatever the problem that you have undertaken, is converted into a valid statement, right? So next slide, please. And whatever that, please go back. Reproducibility is also very, very important. Reproducibility means whatever the work that you have taken, it should be once again, if somebody comes and tries to replicate the same, he should be in a position to get the same results. Then only it is called research. Research means if I do some experimentation, some results are coming, the same procedure, same protocol, if somebody else follows, and if he is unable to get the result, then it is not called as a research, because whatever that we do, it must be demonstrated, it must be reproducible by an established procedure. Whatever the procedure that you are stating and that you are saying that this particular procedure can give these results, means the same if somebody follows, somebody replicates, it should be reproducible. Then only it should be called research, which means first thing you should carry in your mind is systematic investigation, hypothesis, and scientific. It should be reproducible, and it should generate a new knowledge. Then only it can be called as a research. Next slide. Types of research. Lot of research types are there: qualitative research, quantitative research. But let me focus on the types of research generally we follow in pharmacy, right? So basic research is generally conducted on cells and molecules, molecular level research. If you conduct a research on cells, cell receptor signals, receptors, signal pathways, cell membrane physiology, so then it is a basic research, right? So molecules means whatever the molecules that you take. So in chemistry, lot of molecules we synthesize. That is also basic research. Then the main purpose here in the basic research is we only understand the mechanism. We try to develop some theoretical concepts in the basic research. Basic research is all about fundamental research. Fundamental means we take some basic concepts, fundamental concepts, and we try to understand some mechanism. Suppose pain is produced by some mechanism. Nociceptor pathway is the established mechanism so far as regards to the pain, right? So if you want to manage the pain, then do something, manipulate something in the nociceptor pathway so that pain can be managed. Suppose by your studies, by doing some studies, you have come up with a new pathway which is also having some influence on the pain management, then that comes under basic research. So next, applied research. Applied research in the sense, so whatever the research generally in pharmaceutics that people are doing is all comes under applied research. Basic research is generally for pharmacology and pharmaceutical chemistry. Those people will do basic research, right? So coming to the pharmaceutics background people, we generally do applied research. Applied research means already established protocol, established things will be there, like formula, design and development of sustained release dosage forms, formulation and evaluation of buccal release drug delivery systems. So all these things come and just we replace some drug. Only some drug is replaced, entire procedure will be followed same as it is, right? That is called applied research. See here generally formulations we conduct, we do it in the laboratory. So problem solving, whatever the, see every even though the procedure is same, even though the drug delivery system is same, every drug has its own challenging problems, right? Suppose if you want to formulate and evaluate tablets for some drug, let us say, all drugs may not be compressible. Drugs which are having good flow property, drugs which are having good compressibility index, those drugs can only good solid state properties, those drugs can only form tablets. So suppose one drug is unable to be formulated in the form of a tablet. In such situations, that is a challenge, and how to improve its compaction, how to improve its compressibility. By adding some excipients, by adding some other materials, you can improve its compressibility, compaction property, so that material which is actually not possible to be formulated in the form of tablets can also be formulated in the form of a tablet by using some excipients. For this, let me give you one example: liquid solid. So which means generally some substances which are actually deliquescent in nature. Deliquescent in the sense they become liquid in the form the moment they are exposed to atmospheric conditions. Then immediately it will absorb some moisture and it will dissolve. So such type of substances also, for such type of substances also, there are some reported literature saying that they can also be converted into tablets, right? How? By adding some excipients. Drug interacts with some excipients and their solid state properties changes. As a result, a liquid can also be converted into solid. This is a type of challenge and which can be done in the applied research. Next, coming to the translational features. Translational research means all the clinical studies. Whatever the clinical studies that we are doing, lab to clinical, bench to bedside, all those pharmacology related clinical studies, pharmacy practice, where we monitor the adverse drug reactions. So all those studies, they come under suppose if you want to do some clinical trials also, it comes under translational studies. Then preclinical studies, you know very well. Preclinical studies means once the drug is synthesized and it has some potential to show some therapeutic effect or biological effect, then we go for preclinical evaluation. Preclinical evaluation means we evaluate its safety, toxicity and its biological activity. All these things we are going to do as a part of pharmacological studies only, that is preclinical evaluation of some natural compounds or also even synthetic compounds. Then comes clinical. Once again, clinical studies, clinical research means once again evaluating the data. Suppose one drug is pharmacovigilance, they come under these particular clinical studies. Once again, clinical studies are nothing but whatever the drug, how it is going to show its effect on selected population or collective population in general, or in a particular group, like particular age group, in pediatrics, in geriatrics, how a particular drug is going to show its effect, that can be studied under clinical type of research. Now you came to know, apart from this, there are other types of research also, which is called qualitative research and quantitative research. Qualitative research means in this particular research, generally we don't conduct any experimentation. We don't conduct any experimentation, just only surveys by using some theoretical surveys, questionnaires, interviews. We try to extract some information. So that information is called, that particular studies is called qualitative studies, right? So if you administer some questionnaire and get some responses, then that is called qualitative study. Suppose in this particular lecture, I would like to know how many number of students are having above 80% in the last semester. Then I will give some questionnaire, I'll ask you to fill up that questionnaire. Based on that questionnaire, and I will say that this many number of students are having 80% and above. This is a kind of qualitative research, right? So quantitative research, in the sense, in pharmacy generally we do quantitative research. Whatever the laboratory research that we do, whatever the research that we carry out in our experimentation, that is nothing but quantitative. Quantitative means in numbers, right? That is quantitative research. Next, in numbers and quantity. Next. So then what is a research design? Why research design is very, very important? So research design is actually a blueprint. It's actually a particular plan, a particular sketch, right? If you don't have this particular blueprint, then whatever the studies that you design, if your design itself is having some fault, then you can't expect good results out of your research. That's why you should be very careful and ensure that all these elements are present in your blueprint, in your research design. It's a blueprint for conducting research in a logical, valid and economical manner. Not only economical, scientific manner. Logical, valid, scientific and economical. Logical means it should have some logic. Otherwise, suppose you are doing something means without having any logic, without having any need. Suppose some need it should have. Otherwise, why should we design? So we are developing some new anti-inflammatory agents, let us say. Why we are developing, attempting to develop new anti-inflammatory agents? Because majority of the anti-inflammatory agents are causing nephrotoxicity, are causing irritation to the stomach. So these are the problems which you are generally encountered by the already existing molecules in the market, and hence there is a need to go for newer anti-inflammatory agents. So that's why you are taking up a particular research project to synthesize a new anti-inflammatory agent, right? So some need it should have, then that is called logic. Then that is called logic. And next, valid. Whatever that you are doing, it should be scientifically accepted. It should be accepted by all the scientific community. When they will accept your results, when they will accept your research design, when you propose it according to the established procedures and protocols. If you violate all those established protocols and procedures, nobody will consider your research design, and your research design is considered to be invalid, right? Next, scientific means whatever the research design that is there, it should have some scientific elements. Scientific elements means cause and effect, right? If you have taken some problem, so this is the input and this is the output. So how this output is varied by varying the input. So that type of cause and effect analysis, cause and effect relationship should be present in the problem which you have taken. So suppose you see what are the good components in a good research design, what are the components you should have. First thing is you should have objectives. Second one is methodology, sampling, variables, statistical plan and timeline. These are the components which are supposed to be present in the research design. First thing is objective. Objective, how you will come across identifying some objectives. And improve statistical validity and research design. The main purpose of research design is it will prevent the bias. How it will prevent the bias? When you thoroughly conduct the study, when you thoroughly develop a particular research design, so bias means what? Bias means some error. Bias means some error. So if you scientifically design your research design, if you perfectly prepare your design, then there is a less chance of bias. When there is a less chance of bias, whatever the results that you get out of your study are easily accepted by the scientific community. They are considered to be scientifically valid, right? Next. So generally this is the structure of M.Pharm dissertation thesis. So title, you have to first, you should find out your title. What is the title? Title should not be too long. Title, generally people say title should be attractive, but as per my knowledge, title should be meaningful. Title should be meaningful, and it should be as minimum as possible. Not too lengthy, not too short. And it should convey accurate meaning. Instead of attractiveness, it should convey meaning, and it should not create any confusion. Your title is something, your work is another thing. There should be a coherence. If the word called coherence, there should be coherence between the work that you propose and the title. So your title and your work should be in coherence, right? So please ensure that whatever the title that you give to your work actually reflects your work, right? So next, introduction. In the introduction, generally, so we present so many things, but a brief background is enough. Generally in your thesis, you write so many pages, but if you write around 3,000 words, that is enough. For introduction, not in thesis I am telling, but in your publication, introduction should be not more than 800 words. But in thesis, if it comes to thesis, then you can go up to 3,000 words, and that introduction should have background, the related background, related theory, related concept, Suppose some drug is there, why that particular drug is there, some statistics. So whatever the points that you include in your introduction should actually support your title, should support your research work. It should act as a evidence. By presenting those points, it should strengthen your research topic, right? So whatever the statement or whatever the sentence that is irrelevant, that is actually not contributing towards the actual meaning of your topic, please exclude it, right? So generally introduction should be some background, divide them. So brief background you should give, and need of the study, why you are undertaking that particular study, aims and objectives, all these things you should include in the introduction, right? Drug profile, if you are using drug profile also, you should include in the introduction. Then literature review, I am going to discuss about this in detail in the coming slides, right? Next, after literature review, then you come up to you come to the objectives, and then methodology, results, discussion, conclusion, refer. Please remember this particular structure. This is the actual structure, and it is logically connected, step by step. Everything is in sequence, right? First you identify one particular topic, that is converted into title. Then after title you develop some introduction, which means some theoretical work you do. So after you get some introduction, then you go for some literature review. Why literature review? To identify the research gaps, and also to identify to formulate the objectives. Once you get the objectives, how to achieve your objectives. Yes, you have some set of objectives. By what means you are going to achieve your objectives, that is going to be decided by your methodology. What methodology you are going to use, in that materials and methods. What materials you use for the research or your project work, and what methods actually needed to achieve those particular objectives. That should be very clear, and preferably methods should be adopted, imported from standard peer-reviewed journals. Don't rely on the substandard journals methods. Don't rely on substandard journals materials, because it is all cooked up research. It is not a general research. It is not a original research. It is all fake research. If you rely on those particular methods, definitely I am sure you are not going to get the results. At least my suggestion for you is refer to at least 2 and above impact factor journal, not only 1. Why 1 I am telling? Even the manipulated studies, if it is presented very well, can be accepted in 1 impact factor journals, because this is the experience which we are having. We have seen also so many times. Well presented articles, well articulated articles without having any substance in it can be accepted in 1 impact factor journals. 2 and above impact factor is quite good. You can strongly depend upon the procedures, the methods which are mentioned in the research journals or research papers above 2 impact factor. Next, results. Go back. Results. Once you carry out your methods and all those things, you will get some results, some data you get, right? And how you, once you get the data, arrange them in a logical manner, right? So when you arrange them in a logical manner, that is called discussion. That is called discussion. And after discussion, then how you interpret your results. After interpreting and interconnecting your results, you will get some conclusions. That is your final ending of your particular research project, right? So you arrive at the conclusion, from the title to the conclusion. These are the main steps which you are supposed to follow. And don't jump directly into conclusion, don't jump directly into results. So it is all in a sequence. Come in a sequence, then only you will be able to arrive at the conclusion. And references, very, very important. How you follow the Vancouver style or any other style, some standard reference formatting structure is there. Follow that standard formatting referencing structure throughout your thesis. Don't switch on to Harvard style, Vancouver style. In one introduction, Harvard style. In materials and methods discussion, Vancouver style. Generally, people will not accept. I don't know about SPPU general guidelines, which method, which reference style they follow. Please refer to the guidelines and follow that particular style of reference only. right? So people are very crazy about references. Generally you think that people ignore references, but as per my knowledge, majority of the scientifically sound knowledge people, they first look at the references, because once you go through the references, then he will be able to assess whether the research work which is being presented is having some quality or not. After going through some references which are from very substandard sources, then immediately they will throw your research or whatever the thesis that you have prepared. So please ensure that at least you may not be able to do a very high quality research, but we will be able to use high quality references. For that there is no particular limitation, right? So when you refer high quality journal, at least quality of your, that is the first impression. Generally whenever your thesis is sent for evaluation, they look at your references first. If you cite and include standard references, then at least they'll get some positive mindset about your particular research work, right? Is it clear? Reference means impact factor 2 and above. Next. So formulating a research problem should be, most of the times we tend to prepare our research problem as novel as possible. As novel as generally. Is it novel? Is it novel? This is the consistent question which is being asked by so many reviewers, so many examiners. Is it novel? Is it not? But as per my knowledge, is it feasible? First you check out that. It may be novel, but if it is not feasible, then it is not possible, right? First, please ensure that whatever the topic that you have taken, is it feasible, possible or not? Then instead of checking for the novelty, if it is novel, it is well and good. But if your topic is not novel, don't be disappointed. We have other options to present our work. If it is having good application, if it is having good contribution, if it is having good significance, if it is having good relevance, then even if your work is not novel, it will be considered, right? So novel, but it is not at all useful, novel, but it is nowhere used, then nobody bothers about your work. If it is novel, it is well and good, but please ensure that your work is feasible, your work is relevant, your work is application oriented, your work has some significant contribution to the existing field of your specialization. So here, just example I am giving, improving solubility of BCS class 2 drug using novel formulation status. So here, improving solubility so many people have done, but if you come up with a new strategy wherein solubility can be increased, then that is considered to be a good problem. That is considered to be a good project work, because you are using some different method, you are using some different strategy, and that strategy should not be too costly. Ensure that it should be very simple. Ensure that it is very cheap, very economic, without compromising on the quality of the work. Quality of the work is also very important. Next. So writing objectives is also very, very important. Generally people tend to write lot of lengthy objectives. Objectives which cannot be achieved. Please check before you write your objectives, whether I am able to achieve this objective or not. Nowadays people are using lot of ChatGPT, right? ChatGPT, the problem is it will give you some some hi-fi objectives, some objectives which cannot be achieved actually. Such type of objectives if you write, there is a chance of rejection, there is a chance of point out. Then they will understand that this particular person has used ChatGPT. Please learn how to use ChatGPT. Use ChatGPT, but don't reflect that you have used ChatGPT in your work. When you can do that, when you know how to edit the responses that you get out of the ChatGPT. Whatever the responses that you got from ChatGPT, if you include, then people understand that you have copied all the content, generated all the content from artificial intelligence and you don't have any natural intelligence. Use natural intelligence to counter artificial intelligence, right? Next. So just wait. So smart means, so here SMART, you can see S stands for specific, M stands for measurable, A stands for achievable, R stands for relevant, and T stands for time bound. Right? It should be specific, it should not be vague, it should not be general. Right? So how to write our objectives? In the sense, objectives for pharmaceutics different, for pharmacology different, for pharmaceutical chemistry different, and all those objectives I will discuss one by one in the coming slides. Right? Next. So hypothesis development. So here most of the times we ignore hypothesis development. I have seen so many students theses in our, especially in our pharmaceutical sciences, we generally forget about writing a statement about hypothesis. Hypothesis is very, very important. When I was carrying out my PhD, I have included hypothesis statement in my PhD work, right? There are two types of hypothesis, null hypothesis and alternate hypothesis. So alternate hypothesis is accepted when the results are statistically significant, right? When the results are not statistically significant, then you have to reject the alternate axis, which means no result you got. Whatever the assumptions with which you are proceeding is wrong. So if you get statistically significant result, then how to write hypothesis? That is also very, very important. Null hypothesis, alternate hypothesis, right? So here you see solid dispersions does not improve dissolution. Actually your intention is to improve the dissolution. You want to improve the dissolution by using solid dispersions. But to write a null hypothesis for this is you have to write negative statement. So here the negative statement is solid dispersions does not improve the dissolution. This is my null hypothesis. This is the negative statement. Suppose if I get statistically significant results, to reject this particular null hypothesis, then I reject this null hypothesis and accept that solid dispersions improve dissolution significantly. So you should be in a position to write hypothesis statements for your research project. Suppose you are synthesizing some isotin derivatives. Suppose I am synthesizing some indole derivatives, and indole derivatives, why I am synthesizing indole derivatives, I will write something in the title. The design, synthesis and characterization of indole derivatives for some biological activity, Then for this null hypothesis, how I should write? Right? So if for this the null hypothesis is whatever the isotin derivatives that I have synthesized does not produce any biological activity. This is the negative statement. Generally my positive statement was design and develop or characterize, synthesize indole derivatives for some possible biological activity or any anti-cancer activity, whatever it is. Then in null hypothesis you should write in a negative sense. Whatever the derivatives that you are synthesizing does not produce any biological activity. This is null hypothesis. Then the alternate hypothesis is whatever the derivatives that we synthesize do produce some biological activity. When your results are statistically significant, then you can reject null hypothesis and accept alternative hypothesis. Next. So variables in research, very, very important. How to control the variables in research? Because variables, if properly controlled, then you will get the actual results. You should get good control. You can get good control over your results only when you identify the variables present in the study. So variables present in the study, why should we identify, why should we monitor? Because the identified variables are actually contributing for the output of the study. Output of the study is actually dependent on the variables. So what are the variables generally present in the in our pharmaceutical sciences? I have given example for pharmacology, pharmaceutical chemistry and pharmaceutics. So here let me take the example of pharmacology. So independent variables, as the name itself suggests that this is not dependent on anything. Dose of a particular drug. It is in the hands of the investigator. I am the investigator of this particular research work and I want to select some dose, whether it can be 5 mg, 10 mg, 20 mg, depends upon my choice. If the variable depends on the choice of the investigator, then that is called independent variable. So here dose of antihypertensive drug, used in a particular study can be considered as independent variable, right? So depending upon this dose, there is a relationship, there is a direct response connected to this particular dose, that is called BP, blood pressure, hypertension, systolic blood pressure, diastolic blood pressure. So if you observe reduction in systolic blood pressure or elevation in the systolic blood pressure, then it is called dependent variable, because these changes in the systolic blood pressure actually attributed to the dose. So that's why that becomes independent variable, this becomes dependent variable. So if you want to vary something in your studies, vary some whatever the systolic blood pressure in your study, then try to change independent variable so that dependent variable can be changed, right? You can't actually directly change systolic blood pressure. Systolic pressure you can't change directly because it is a dependent variable. Identify the independent variable on which this particular variable is dependent, then you can vary it. So next coming to controlled variables. So controlled variables are very, very important in a particular study setup. Okay, to assess the actual relationship between dependent variables and independent variables, you should take into the consideration of controlled variables and confounding variables, right? If you don't pay attention to these two variables, even if your study is correct, even if your intention is right, even if your setup is proper, you may not be able to get the results. We blame some research papers. Hey, this particular person has given wrong method. I'm not able to get that particular result. The reason is you might not have paid attention towards controlling variables and also confounding variables. You have to pay attention here. You have to identify what are the controlled variables and confounding variables in your study and keep it in a minimum, keep it at an optimum level. Then you will be able to study the relationship between dependent variable and independent variable. So here salt intake, age group, measurement. Suppose salt intake is very high, right? Then it contributes to the elevation in the systolic pressure. Then you can't attribute that particular change to the dose of the drug. So here since you have not controlled this, you may not be able to come out with the relationship between dose of hypertensive drug and also its response, right? So next, confounding variables. Confounding variables are plenty in case of pharmacological, in case of clinical studies. So stress levels, smoking habits, other medications. Suppose candidate is using other medications, then this effect, whatever the effect that you are seeing, cannot be directly interpreted, right? It may be because of other reasons, right? So in your study, identifying these four variables is very, very important. First one is independent variable, dependent variable, controlled variable, and confounding variable. Confounding means bias, error, right? Next. Now this is an example related to pharmaceutics, right? So in pharmaceutics, once again, independent variable stands for percentage of HPMC polymer that you have used. Why HPMC polymer is a independent variable here? Because the concentration of HPMC polymer that can be included in the formulation depends upon the choice of the investigator, choice of the experiment. It is my wish. I want to include this much HPMC level, this much concentration of HPMC. That is according to the interest of the experimenter. So based on that, this particular percentage drug release is going to be changed. So why percentage of drug release is changed? Because polymers are generally known to retard the release of the drug. Whenever you add some polymers, what polymers will do the formulation? So it will control the rate of drug release. So rate of drug release controlled by polymer actually depends upon nature of the polymer and also concentration of the polymer. Nature of the polymer is not in the hands of the experimenter. That's why it does not come under independent variable. Nature of the polymer is not in my hands, but what is there in my hands? In my hands, concentration of the polymers. So hence it is a independent variable. Next, dependent variable is percentage of drug release at so and so hour. Controlled variable, once again, tablet hardness, dissolution medium, RPM. These are the things you have to control. Otherwise, you can't establish the relationship between concentration of the HPMC polymer used and the percentage of the drug that is released. Right? So its relationship depends upon certain conditions set at control variables. At particular, at this much of tablet hardness, this will be the relationship. If hardness changes, that relationship also changes. At this particular, in this particular dissolution medium, suppose acidic dissolution medium, that is 0.1 normal HCl, one type of release, 6.8 basic pH, another type of release. So here the release is not related to the actually concentration of HPMC polymer and release of the drug, but it is related to dissolution medium. Hence, keep it at a constant level, control it, so that you can establish the relationship between the level of the HPMC polymer and the rate of the drug or amount of the drug that is released. Next, confounding variable here is granule sizes, moisture content variation, other things. They should be kept at minimum level, so that whatever the independent and dependent variables under the study that you are keeping, so they can be actually studied, they can be actually related and correlated, right? Next. So this comes to pharmaceutical chemistry. An example for why I have taken 3 examples, because we have 3 types of background students: chemistry, ecology and pharmaceutics. Now the same can be applied to chemistry also. So here reaction temperature is in our hands, and reaction temperature and also the concentration of the reactants, here I have not written, but concentration of the starting materials, in what molar proportions you are adding that material into the reaction mixture actually influences your yield. What type of catalyst you have used, what is the concentration of the catalyst that you have used, what is the temperature that you have used, actually influences yield. So yield is a dependent variable, and to increase the yield, these are the independent variables which are to be monitored. And in that, control variables are reaction time, what type of solvent you are using, molar ratios of the reactants. So if you change the molar ratios, how the yield is changing, right? So then confounding variables is, if your reagent itself is having impurity, then you can't attribute that particular yield changes to the neither to the reaction temperature nor to the molar ratio, because there is a fault in your reagent itself which actually spoiled your experiment. And don't blame the either the reaction temperature or the conditions of your reaction, that is solvent and reaction time, but here the problem with impurities in your reagent. That's why whatever the materials that you use in the study, ensure that they are having good quality. In case of pharmaceutics, generally whenever we use the particular drug to incorporate in the formulation, we take a DSC. Why DSC, differential scanning calorimetry? To ensure that it is pure. If it is impure, then you will get 2 different peaks. One peak you got means only one substance is present in that, then that can be safely used. If your drug itself is impure, then it will amount to lot of inaccurate results. Okay, so purity of the reagents, purity of the raw materials, or the materials that you use, or the equipments that you use, conditions of the equipments that you use actually contribute towards the quality of the results that you get, right? Next, confounding variables, impurities in region, atmospheric moisture, inconsistent steering. These are all the things that are there in particular in the pharmaceutical chemistry. Next. So now we will come to the specialization specific concepts, right? First, let me start with the pharmaceutics, right? So example project here is formulation and evaluation of solid lipid nanoparticles of curcumin for enhanced oral bioavailability. Enhanced oral bioavailability means the moment you look at this particular title, your title should be in such a way that the moment you look at this particular title, people should be able to guess the problem, people should be able to guess your methodology, people should be able to guess what you are going to do. If the people are comfortable in doing all these things, then you can say that your title is perfect. You can say that your title is actually carrying your intention and meaning, right? So here problem is, because why you want to enhance the oral bioavailability? Because it's poorly soluble. Problem is curcumin has poor solubility and hence poor bioavailability. So here dependent variable is bioavailability. Bioavailability is dependent variable. Independent variable is solubility, right? In between you have another controlling variability or confounding variability is permeability. metabolism. These are all controlled variables and confounding variables, right? If you don't monitor those things, you can't expect a direct relationship between solubility and bioavailability, right? Solution is develop solid lipid nanoparticles to improve absorption. Why solid lipid nanoparticles? Because solid lipid nanoparticles improves the absorption. When absorption is improved, bioavailability is improved, right? So next comes literature survey. So in particular, generally, whatever the research problem that you have taken up in the last slide, the research problem which we have taken up is developing solid lipid nanoparticles for curcumin. The problem here is curcumin has poor boral bioavailability, poor solubility. These are the problems, and these problems must be identified, and these problems must be addressed by using a novel formulation strategy called solid lipid nano formulations, right? So to develop this, first we should go for literature survey. Literature survey means, is there anyone who has already developed solid lipid nanoparticles for curcumin? First question. Yes, there are some people who have developed solid lipid nanoparticles. Don't give up. Even if they have developed solid lipid nanoparticles for curcumin, you have something to add for that. Suppose they have used some X lipid and you screen for another lipids. So and so lipids, so and so particular people have reported curcumin nanoparticles, solid lipid nanoparticles with so and so lipid. Then screen for another lipid. All lipids are exhausted. Suppose all lipids are used by all, and no lipid is left behind which can be used for solid lipid nanoparticles. In that case also, don't get disappointed. You have another choice where you can do some modifications in the methods. What methods they have used? Is there any new method which you can use to go for solid lipid nanoparticles, right? So there, if you are developing a new method which other people have not developed, then you can go for executing that particular project, right? But don't repeat whatever the people that have they have done. Already they have used some lipids, already used drugs, already used methods. There is no sense, no meaning of doing research. In fact, even if they have done all those things, there is one more thing that you can do. Suppose they have evaluated it for some disease or some particular activity, then you can go for another activity. Suppose solid lipid nanoparticles of some X drug has been developed and its pharmacological activity has been evaluated in so and so conditions for about, so let us say for anti-hypertensive activity, suppose, then you can go for anti-cancer activity. In that way also you can show some variation and show that your project is not a duplicated project. It is a project which is having some distinct contribution to the scientific community, right? Now coming to the literature survey, how you are supposed to do the literature survey. So there is a systematic research that is presented over here, right? In the suppose same drug, same topic, solubility enhancement techniques for BCS class 2 drugs, let us say. So this is our topic, and for this topic, how to carry out literature survey, right? There is an established procedure called PRISMA, preferred reporting of items, systematic reviews and methods, meta-analysis. This is the expansion of PRISMA, right? So in this, first what you are supposed to do is search some standard sites, standard search engines. Standard search engines are PubMed is there, Scopus is there, and also please select some duration. Duration is also very, very important. So suppose 2000 to 2025. In this duration, whatever the articles which are related to this particular solubility enhancement techniques of BCS class 2, first identify them. Just identify them. So suppose, let me tell here, they have identified around 1500 papers. 1500 papers, if you identify, then you are a very great person, right? Generally it is given like this, but for PhD, it is advised to go for this much of extensive research. Extensive literature survey is needed. If you go for PhD work, then at least you should go for, here it is mentioned 1500, but I am telling you at least 500. If you refer, it is well and good for a PhD work. For M.Pharm work, 200. Refer at least 200 relevant related articles. Related references you should first identify. Identify its source, title, where it is published. Where it is published, you just identify. Then, after identifying, then you should go for filtering them, right? So, filtering, how you filter that? So, remove the duplicates. Remove the duplicates means on same drug, same article if they are published, just exclude that. Same method, exclude that. Right? Same technique, exclude that. Same characterization, exclude that. Same evaluation, exclude that. So, keep some exclusion criteria and narrow down your research papers. From 200, come up to 100. From 200, exclude 100 and then come up to 100. Then, screen for titles. Screen for titles which are actually relevant to your work. Whether these titles are actually reflecting the meaning and idea of your work or not. So from 100 you come up to 70, 50, right? Then 50 full text articles you should have. 50 full text articles you should have. Thoroughly examine for its objectives, thoroughly examine it for whatever the methodology that is being used and how the results are reported, what are the different equipments that are used for analysis. All these things you have to critically examine, critically evaluate. Right? Next. So here, this is how you have to prepare a table. Literature survey means don't do a kind of what you call without any logic. Don't do the research literature survey. Literature survey itself is a important activity which is there in your research work. If you do thorough research work, then you get lot of ideas. Actually, the input for any research is a research literature survey, right? If you want to get some good idea about your research, or if you want to come out with a solid research problem, then you have to refer to the literature thoroughly and critically evaluate all the research and prepare a table out of the whatever the journals that you have searched, right? Suppose this is the table that you can include like this. See, 50 research papers you have studied and 50 research paper, prepare a table for that, so that you will easily get idea, command over your research topic. If you want to develop command over research topic, then this is the best method to prepare a table, and in that table, who are the authors, in which year it is published, and what drug it is studied, right? Then which method they have used, what are the key findings, what are their limitations. See here, curcumin, they have developed nano formulations. Bioavailability is improved by 3 times in rats or in animal models, but here limitation is they have not done any clinical trial. It is not yet proved in the human beings, human subjects, right? So next, Rao et al, they have taken metformin drug, they have prepared solid dispersion, they have improved the solubility, but their problem is stability. There is no stability at all. Stability data is missing. There is no stability data which is present in that particular paper. Hence, you can work on the stability data. So that's how where the research has been stopped, there another research starts. Somebody's ending is your beginning. Your ending is somebody's beginning. So that is how the research is connected and continued for generations together, right? Now, here let me present the sample research objectives for pharmaceutics subject. So what are the topics that we have taken? To prepare solid lipid nanoparticles by high shear homogenization, right? So here, first you should prepare the solid lipid nanoparticles, means here they have written like this, but prior to that, first identify another objective. Another objective I am telling, identify which particular solid lipids are actually suitable for preparing solid lipid nanoparticles of the drug which you have taken for the work. Identify those particular solid lipids and check the compatibility with that. So here compatibility studies they have not given in the objectives, but I am telling you, right? So it is generated by ChatGPT, that's why they have not given, but I am telling you, right? So you add some what you call compatibility, whatever the lipid that you have taken, whether it is compatible or incompatible with your drug. First you should check it, right? After doing everything, evaluation, later you found that your drug is having incompatibility issues with your lipid that you have taken, then entire research work boomerang. Entire research work is spoiled. Then you don't get any good ideas, right? So you, whatever the hard work that you have done is of no use, right? So whatever the things that are likely to influence and likely to negate and likely to impact your research work, identify, do it immediately, right? Then that is your first objective, right? So after that, they have prepared some solid lipid nanoparticles. Then particle size should be optimized. How particle size is optimized? By reducing, by adding some variables called whatever the emulsifiers that are there. That emulsifier concentration, if you change, particle size can be changed. Next, evaluate drug release. Depending upon size, once again release also influences. Then assessing the stability, because in formulation stability is as important as safety and efficacy. In our pharmaceutics, stability is as important as safety and efficacy. Generally, any formulation that we prepare, first we check it for its safety. First is safety. Second one is efficacy. Third one is stability. Last one is whether it is economic or not. That is also very important. So these are the 4 criteria by which we evaluate any particular dosage form that is likely to be developed, right? Next. So once again here variables, just now I said variables comes under experimentation part. So if you identify the variables, then the chance of getting quality results is very, very high. So whatever the results that you get, nobody is going to comment on that. Nobody is going to express a doubt on that. So if everyone wants to accept your results without any doubt, without any bias, there should be a proper planning of your experimentation protocol. And proper planning of experimentation protocol depends upon identifying the variables. So in this particular study, variables, so factorial design, what type of factorial design here? Nowadays, no trial and error based studies are accepted. Please remember, for pharmaceutics students, I am telling, if you want to go for any formulation, please include factorial design in your studies, because no trial and error experimentation is accepted. In those days, when the factorial design software were not there, people have accepted, but now everything should go from this particular factorial design experimentation. The design, even for method development also, people are using factorial design. So even for synthesis work also, people can use factorial design. Even for synthesizing some new derivatives also, you can identify dependent variables, independent variables, and develop a particular protocol, trial runs, and accordingly you can go for reactions. That is also a quality study, good study. If you design a study, synthetic study, by including factorial program into that, factorial design into that, then it is going to be a valid, scientifically sound experimental design, and you can justify your results very firmly by using statistically significant results. So here variables, so here factorial design means 2 to the power of 3. So here please understand that in factorial designs, this one stands for base, 2 stands for levels. 2 stands for levels. How many levels we have? Higher level, lower level generally we take, and 3 stands for number of variables. If number of variables only 2, then fine. 3 generally at M.Pharm level, don't go for variables beyond 3. It is very difficult to control variables. It is very difficult to control. Suppose lot of variables are there, right? Lot of variables are there. In such case, how to study variables? Keep some variables controlled. That's why that is called controlled variable and confounding variables. Keep some variables in a controlled manner, in a constant manner. Make it constant. Make some variables constant. Suppose temperature can be constant, temperature can also be a variable. If you keep temperature constant throughout the experimentation, it becomes constant. If you change the temperature throughout, it becomes variable. So variables, constants depends upon our choice. Which variable you want to keep it as a variable, Which variable you want to keep it as a constant, it is your choice, right? So here lipid concentration, surfactant concentration, homogenization speed, these are the variables, and how these variables are influencing the outcome, that is called this is independent and this is a dependent variable, that is particle size and entrapment efficiency. Why these particular dependent variables have selected? Because go back, because here your objective is to optimize size, to achieve that particular size, to achieve the preparation of solid lipid nanoparticles, then you have to go for the change in the concentrations of lipid. Next slide. Lipid and surfactant concentrations and all those things. Next, formulation methods. So formulation method should be mentioned very clearly, right? Step by step, without any confusion. Clarity should be there, right? And it should be in a sequence. First, melting is done. Melt the lipid. Suppose dissolve the lipid and then melt the lipid, right? It is wrong. First, you have to melt the lipid, and in which you have to dissolve the drug, right? So how systematically, stepwise you are proceeding in your research, same thing you should write in your methodology, right? So that sequence should not be disturbed, right? Each and every step should be preceding step, following step. What is the preceding step? What is the following? Next step, previous step, next step. So if you miss the order, then people will not be able to reproduce your work. To ensure that the other people reproduce your work, you should present your methodology in a correct manner, in an order manner, in a sequence manner, in a logical manner, right? So melt the lipid, dissolve the lipid, then add the surfactant solution, then homogenize it, cool to form solid. When you cool, when you decrease the temperature, the melted lipid will become solid and it will come out as a solid lipid nanoparticle. Next. Then comes characterization. So here in the characterization, particle size, zeta potential, entrapment efficiency, images, surface images, in vitro drug release, and in the in vitro release studies, once again dialysis membrane, what type of apparatus you are using and how you are modeling that particular release kinetics, all these things you have to write. And here another important thing is statistical plan. What is, if you use design expert software, that software will give you statistical significant results or not, it itself will tell you. But if you don't use that, you can also use ANOVA, response surface methodology, and design expert 1 and 2. Next. Stability study. So this is the stability study. 60% RH, 25 degrees, 40 degrees. Why 40 degrees, 25 degrees? Because this is the upper and lower extremities of our region, right? 25 and 40. Next, duration is 3 months and reduced particle size 200. So this is the plan, right? Timeline of your research work. Timeline of your research work is in the first month literature review, third month only pre-formulation, because to develop a robust formulation, first you should do some preliminary studies which is called pre-formulation. Then formulation, after that formulation, evaluation, you get some data, then analyze it, then thesis writing. So this is all about pharmaceutics research work. Next. Coming to the pharmacology, so here just example project which we have taken here is evaluation of neuroprotective effect of herbal extract in streptozocin induced diabetic neuropathy in rats. So here, after looking at this particular title, first you should get diabetic neuropathy in the rats, right? You should develop diabetic neuropathy in the rats. If you don't develop diabetic neuropathy in the rats, how will you evaluate the effect of neurodegenerative effect or neuroprotective effect of your herbal extract, right? So to evaluate the neuroprotective effect of your herbal extract, first you should induce diabetic neuropathy in your rats. And how you induce that diabetic neuropathy? By following which method, by following which procedure, by adopting method from which article, right? That you should highlight, that you should mention in your research plan. Now here background is diabetes causes neuropathy, oxidative stress major mechanism. So neuropathy, since diabetes is caused by neuropathy, supplementing the patients with some herbal extract so that their nerve cells can be protected. This is the main logic involved in this particular study, right? Next, research object is first induce diabetic neuropathy. If you don't induce diabetic neuropathy, you can't evaluate your herbal extract and you can't say that your herbal extract is protecting the neurons, right? Next, administer the extract, then evaluate behavioral parameters, and also not only morphological behavioral parameters, but you should also have some data regarding the biochemical parameters, because biochemical parameters is the strong data, right? In data, once again, two types, right? Data, quality data, quantitative data. Qualitative data is not that much strong, but quantitative data is very, very strong. Sometimes qualitative data, quantitative data, if you correlate, then it is very good. That is what pharmacokinetic and pharmacodynamic studies. Pharmacokinetic and pharmacodynamic interaction. PKPD modeling. PKPD modeling means pharmacokinetics comes under qualitative and dynamic is qualitative, and if you correlate together their interaction outcome, if you assess, then it is a wonderful study, right? How you are going to correlate pharmacokinetic data with pharmacodynamic data, that is nothing but qualitative and quantitative data interaction. Next, interpretation. Next, experimental design. So in pharmacology especially, this design is very, very important, right? If your design is wrong, then your outcome is wrong. How many groups you are forming, right? Control group, naive group, normal group, diabetic control, standard, test, low dose, test, high dose. How you are grouping the rats? How you are grouping the animals? On which animal you are actually testing your extract? That is also very, very important, right? So here animal model is rat, they have taken. So streptozocin, 55 mg interperoneal, all these things, and behavioral studies, heart plate test, tail flick test, rotarat test, these are all the behavioral studies, right? Qualitative data you get, and you can record some images also, videos also, to prove that whatever that you are doing is actually true, right? The main intention here is whatever the results that we are presenting in our thesis, what is your burden? The burden of proof actually lies with the experimenter only. You should prove that whatever the results that that are produced in your research work are true. For that, what evidences, what undisputed evidences you can produce, you should think of that. Even videos can be manipulated. Nowadays people are saying, by using AI, even videos can be manipulated. In such case, what can be the best way of presenting your results without any bias, without any doubt, right? That is also, you can think and you can innovatively present your own results. Next, MDM, malondialdehyde and superoxidase dismutase catalyze TNF alpha. These are all the markers which play a role in the neuroprotective mechanism. MDA SOD actually responsible for oxidative stress. Oxidative stress, if it is decreasing, means the herbal extract, if it decreases oxidative stress, means, because in the first slide itself I told that what is actually contributing for the neurodegeneration, diabetic neuropathy, oxidative stress. So if your extract is able to reduce the stress, then you can conclude that this extract is having neuroprotective effect. So next, histopathology, nerve sections you can take, and if it is neuroprotective, before the administration, after the administration. Before the administration there is a nerve damage, after the administration the nerve damage has been recovered. Such type of histopathological studies also serve as a solid evidence to prove your thesis statement, right? Next, H&E stainings, and for this ethical. So in the beginning itself, your research problem should be ethical. Don't do unethical research, right? Unethical research is not at all good. I will tell you what is unethical research, especially for organic chemistry people. Those who know good synthetic, those who are having good synthetic knowledge, they are hot cakes for whom? For narcotic mafia. For narcotic mafia, those scientists or those organic chemists who are having sound knowledge in synthetic design and synthesis of various derivatives, they will be easily trapped. They say that you synthesize this derivative, this morphine derivative, this cocaine derivative, for which we will give you crores of money. Then if you take up that unethical research and do work on how to improve the yield of these narcotic drugs, then that is unethical. It may be highly lucrative in terms of money, but it is, for society, it is unethical. So don't take up such type of unethical problems, right? Next. Next comes for pharmaceutical chemistry people. I have not done anything new. Whatever the research work that I have done, I thought of showing to you so that you will be able to understand from that. Next. Next. So I worked on indole derivatives. So for this, first you should present whatever the, suppose you are working on so and so nucleus, right? How that nucleus actually come into existence. How nucleus of indole is being formed. That I have presented in the first slide. Evolution of, so this comes under introduction. This comes under introduction. In the introduction, brief history, brief evolution, some interesting facts, some statistical data, some points which can actually stimulate the intellectual discussion that you can add in the introduction. Next slide. Next slide. Next slide. So marketed drugs with indole nucleus. Suppose you are working on solid lipid nanoparticles of some particular drug. What are the other formulations available in the market on the same drug? You can add and you can present in the research work. Next. So what is isotin? Its chemistry, especially for chemistry people, discussing the chemistry of the nucleus is very, very important. Chemistry of the nucleus in all domains, right? Next slides you can see. Next sources, from which source that particular nucleus can come, because any compound, it can have plant source, animal source and synthetic source, right? So from various sources you can isolate that particular compound. That also you should give. Next. Synthetic roots. What are the synthetic roots available to synthesize that particular nucleus, and which synthetic route you are using, that justify why you are using that particular synthetic route. Generally, the choice of a synthetic route depends upon yield. Number one, high yield. Number two, impurities. You should get pure drug and no impurities. Number three, number of steps, number of reaction steps. Generally, we aim for single step reactions. Single step reactions are gold standard reactions. Suppose in your project work, to show that you have done lot of work, you have developed so many multiple synthetic reactions, then people may not look, or people may not get impressed at your work. Suppose some multi-step synthesis is there, that by using your logic, scientific knowledge, you convert it into single step reaction, then that's a great contribution. Suppose low yield drug is there, you have proposed a new alternative, synthetic alternative, great research. Suppose raw material for that particular drug is very economically not friendly, suppose ecologically toxic, suppose not economic, not eco-friendly, but you have come up with another raw material which is eco-friendly and economic, then your project is very good, right? So this all comes under process chemistry. This all comes under process chemistry optimizations. So next, reactivity of isotin. So whatever the nucleus that you take, you should present its reactivity, right? By where exactly the reactions actually take place on that particular nucleus. So here we have three different sites here. This is one site, and aromatic site. In all these sites are accessible. All these sites can be substituted, but the conditions actually vary. If you change the reagents, then the kind of substitution is going to change. If I want to operate or substitute, if I want to substitute one particular chemical on aromatic nucleus, then your choice of ingredients, reagents and conditions will change, right? Next, N-alkyl substitutions, C2 substitutions, then next slide, C3 substitution, aromatic substitution. So these many substitutions are possible on this particular indole nucleus. On the selected nucleus of your research work, identify where exactly it is going to react. What are the possible sites present on your nucleus, and how those nucleus is actually going to contribute for developing newer derivatives. Suppose in the literature it is understood that all these here total 4 sites are there. After conducting thorough literature survey, you came to know that this particular site is accessed, this is accessed, this is accessed, but this is not accessed. Using this particular site, no derivative has been prepared, let us say, then that is a good scope for your research work. Like that you have to identify. Like that you have to identify whether all the sites available for reactivity on the nucleus have been exhaustively studied or not. If they are exhaustively studied, then you can go for another type of problem, right? So suppose already all sites have been studied, all sites have been already reported, then you can think of different type of derivatives, different type of electron withdrawing groups, electronic donating groups. Generally derivatives are planned based on electron withdrawing and by varying electron withdrawing groups, by varying electron donating groups, and also length of the chain, carbon chain length, because these are the independent variables which actually influence the dependent variable called biological activity, right? And also a dependent variable called stability. Stability is also influenced. And for chemistry students, my sincere advice is, please don't play with the chiral molecules. If your molecule is chiral, exclude that. It becomes very difficult for you to go through that particular molecule. Very difficult to handle, very difficult to characterize, very difficult to identify. A lot of questions are going to come up and you may not be able to answer that. At the level of M.Pharm, please don't take up chiral molecules. If we have a great infrastructure by having chiral chromatography and other things, then you can consider, but here we have only one chromatography, HPLC is there. So for that, please don't look at chiral molecules. Simple molecules, synthetic steps should be simple, significant, only single step reactions. Try to aim, focus, so that you will be able to get easy results, immediate results, significant results, right? Next. So different types of synthesis, how the synthesis is proposed, how synthesis has been done, you have to mention the synthetic routes. Next. Go back. So this type of data, statistical data, actually creates some interest, and excitement. So never and ever miss this type of slides in your presentation. Add some statistically significant data. Suppose here research articles and patents on isocyanates derivatives. You can see that was a work which I have presented in 2014. I have not edited that because only 2 years recent at that time. 2012, up to 2012 I have corrected. This was the work which was presented in 2014, right? Up to 2012, number of articles were 245, patents were 36, which means number of articles and patents are increasing year by year. It means people are taking up this particular isocyanates derivatives, endo derivatives for research work because they are potent molecule, they are biologically active molecules. There is a scope for commercialization. There is a scope for getting a new drug out of this particular nucleus. That this evidence shows that, this data shows that the nucleus which we have selected is actually a hot cake for scientists. Next. And another data, this shows that India is the topmost country which is doing extensive research on endo derivatives. So if your nucleus, you just make a study, conduct a thorough research, whether on your nucleus, our country or which country, or else you can present a simple outlook overview on this nucleus. Western countries, how many studies have been done? Eastern countries, how many studies have been done? India, how many studies have been done? Just prepare a number and present. That will be a new addition. That will be a good presentation. Examiners will get impressed out of that, right? Next. So try to find out the justification for your nucleus. Whatever the nucleus that you have selected, for that, why you have selected that particular nucleus, you should present the justifications. Next, literature review. So in literature review, generally for chemistry, no need to review each and everything. First look at the derivatives. And in the derivatives, what groups they have changed. Whether they have changed electronic donating groups or electron withdrawing groups. In which manner they are proceeding. Identify the compound which is biologically active. So if it is biologically active, what is the discussion that they have written and why it is biologically active, why the other derivatives are not active. which method they have used, which synthetic protocol they have followed. These things you have to look upon and prepare a short note out of that and prepare a table, right? Next. So here I have just where exactly the derivatives have been prepared. At this particular site the derivatives have been prepared, and these are the functional groups which are being altered, electron donating groups. These are electron withdrawing groups, electron donating groups. Okay, on one side, generally in chemistry they compare with electron donating and electron withdrawing. They prepare derivatives and compare with electron withdrawing, how the activity is changing, electron donating, how generally it's a contrast activity. If electron withdrawing groups increasing the activity means electron donating groups will decrease the activity. It is all availability of electrons. Once again, I am telling, availability of electrons on the nucleus actually influences the biological activity. Availability on the nucleus, availability of the electrons on the nucleus actually influence the reactivity. There is no availability of the electrons on the nucleus, then nothing can come and bind to that, because reactions is all about sharing the nucleons, sharing the electrons. Whatever the electrons, free electrons available can be shared so that some bond can be formed, right? Bond in chemistry is, in synthetic chemistry is covalent bond. In biological environment it is hydrogen bond. Suppose you got a molecule, I can easily guess whether it is able to bind to the receptors or not. Just simply check for its hydrogen bond. Simply check whether hydrogen bonding capability is there or not. That is why in our co-crystals in pharmaceutics, all the majority of the drugs, they can be formed co-crystals. Why? Because they will have the ability to form hydrogen bonding. Without hydrogen bonding, there is no biological activity, right? Peptide bond, another bond which is very, very important. Peptide bond is very important for biological activity, right? Because majority of the proteins, amino acids, neurotransmitters, they have amine group, amide group, right? Right? So amide group is very, very important. Amide group is very, very important. Biologically active functional group is. So generally, the medicinal chemistry people, what they do is they ensure that in their molecules amide group is present, because amide group actually reflects the endogenous substances, and majority of the endogenous substances are having amide group in it, right? Next, structure activity relationship also you should establish. You should have strong command on structure activity relationship. If you don't have strong command on structure activity relationship, then you can't develop your derivatives. Then you can't justify the derivatives which you have prepared, right? Next. Next. Just wait. So here you see how simply we have expressed the literature survey. What is the screening study? For which activity it is being done? Anti-breast cancer activity. Bas. पूरा पढ़ने का जरूरत है. No need to read the complete research paper. Sometimes just reading the abstract itself is enough. By reading the abstract itself, you will be able to know whether I should read its full text or not. Most of the times. Just now I told 200 research articles you have to download. You have to actually identify. In that, just by looking at the abstracts, you can eliminate 100 articles. Looking at the abstract itself. Then which cell lines? If it is anti-breast cancer activity, whether it is done on the animals or cell lines, it is done on the cell lines. Next, what is the standard drug they have used? By using which drug they have compared? Okay, generally for any study to report anti-cancer activity, they use some drug, some standard drug, that is doxorubicin. Why? Because recently we have observed World Cancer Day. In that, I asked a question: which drugs are in use for cancer therapy at Anand Rishi Hospital? He said doxorubicin for breast cancer therapy. So doxorubicin is a gold standard which is available in the market and used even till today, right? Now results. In the results section, 4B, 4D, 4G, these are the compounds which are found to be biologically active and they are active against the cancer. That's it. There ends the literature survey. No need to write each and every point. If you identify these 3 parameters, 4 parameters, enough. which biological activity they have attempted, what are the derivatives they have used, and which derivatives actually is having good activity, at what cost. Here I have not written the concentration, but concentration is also very, very important. In what concentration, in what molar range, right? Concentration is also very important. Derivatives concentration, concentration of doxorubicin and concentration of the derivatives. If you add that point also, it becomes very easier to compare, right? Next. See this one, next, that is the identified compound. Next. Combination therapy and molecular hybridization is the concept which we have explored in those days in my PhD topic, right? Next. So go back. So this is how you have to present the literature survey. Simple, name of hybrid, biological activity, reference. Enough. Next. Aims and objectives. Now comes very important, writing aims and objectives. Next. The present work was designed and classified into isotyne pomalidomide conjugates we have to synthesize, mesalamine, benzo, 3 conjugates we have prepared. Okay, specific aims and objectives I will present in the next slide. So first aim is literature review on the synthesis and pharmacological activities of indole derivatives to identify novel strategies in the development of new indole derivatives. Second one is synthesis and characterization of isotyne drug conjugates. In that, 3 different types of conjugates. Next, pharmacological and toxicological evaluation of the synthesized compounds. Then identification of the pharmacologically active isotyne derivatives for further studies. These are the simple objectives. Next. Coming to the specific objectives, synthesis of pomalidomide conjugates, isotyne conjugate, mesalamine and benzoic acid conjugates, and then another objective you can include in your chemistry work is purification of intermediates and final compounds either by recrystallization from suitable solvents or by chromatographic techniques. And within this also, if you do purification by recrystallization, then it is well appreciated. Why? Recrystallization is very simple and very effective, can give you 99% purity. But in chromatography, laborious, hectic, and use of lot of solvents, consumes lot of time. Hence, search for those type of recrystallization, purification techniques where recrystallization is possible. Next, characterization of the all new compounds including those intermediates by physical, analytical and spectral data. Physical means looking at the color, texture, whether it is solid, whether it is yellow, blue, green, texture is semi-solid, rough, all these things you can write. That is physical data. Then acute toxicity studies, synthesis compound, then pharmacological evaluation. Next. See, I have written the hypothesis statements in those days. This was a presentation presented in 2014. I did not do any editions for that as it is because time was very less. After completing pharmacology, time was 3:10. Then immediately, whatever the previous slides which I was having in my PhD presentation, then I extracted, cut and pasted over here, right? So here, research question and hypothesis. The question here is, does the combination of 2 bioactive moieties into a single hybrid moiety produces the additive, synergistic or no effect on the intended biological action? That was the question. I have taken some nucleus called isotyne nucleus and I have added it to the existing drugs, commercially available drugs. Pomalidomide is currently under treatment and second one is mesalamine is currently under treatment. Only benzoic acid is it's a kind of preservative, right? So this pomalidomide, if you add it with isotyne, we observed that its dose has been reduced. The API pure pomalidomide dose is high, but if you add it with isotyne, and we have even checked, right, physical mixture, bonded mixture. If you combine pomalidomide plus isotyne as a physical mixture as it is and observe it on the cell lines, its activity was less. And conjugated with the help of covalent bonding as a single moiety, its activity was good, right? So that was the question. Does the combination of 2 bioactive moieties into a single hybrid produces additive effect, synergistic effect or no effect on the intended biological action? Intended means it can be anything. cancer activity, ulcerative activity, whatever it is. So the question should be as general as possible. Otherwise, the 3 categories of compounds would not fit into a single question. Now 3 categories, 2 are anti-cancer categories, 1 is anti-ulcerative colitis category. But still, they got fit into the research question. Next, coming to null hypothesis. Now, null hypothesis, how we will generate combination of 2 bioactive moieties into a single hybrid moiety produces no effect, because it's a null hypothesis, produces no effect on the intended biological actions. Then alternate hypothesis is combination of 2 bioactive moieties into a single hybrid moiety produces additive, synergistic and on the intended biological action. This is the, please include this particular slide in your presentations, research question and null hypothesis and alternate hypothesis. Next. Once again, variables in the study. Very important. Variables in this. Here also, we identified independent variables and dependent variables. Independent variables is nature of the combined bioactive moieties and concentration of the hybrid moiety. Right? Why I have included nature? Because I am having the choice to select whether isotin or imidazole or any other nucleus. That nature, it is there in my hands. That's why I included it in the independent variable. Right? Then another choice is at what concentration. In my own hand, how much concentration I can use. That's why it is a independent variable. And depending upon that, changes in biological activity take place. Next. Next. Next, go back. Next. See, this is how you have to present whatever the materials and methods that you have presented, you have used, you should present like this. Next. Methods also, present methods for chemistry methods, follow this type of practice, right? Make a square, that square stands for a reaction vessel, right? So this type of block diagrams actually gives a lot of clarity, very appealing, easily you can understand the procedure, right? The square box which is designed actually represents reaction vessel. It can be RB flask, anything. Now one arrow, first arrow comes, first arrow, next. First arrow means number 1 addition, second arrow means number 2 addition, third arrow means number 3 addition, then finally, so all these input materials reacts inside and will give you an output called isonitroso-acetone. So if you present it like this, if you present your reaction procedures in this manner, it will be very easy to understand. Very easy and elegant, and examiners will get easily impressed out of your presentation. Next. Go back. Next. Results and discussion. Next. See here, this is how you have to tabulate. This is the physical data. Molecular formula, melting point, yield. You can also add some color. What is the texture? Whether it is solid or liquid. This I have not added here. But generally, melting point, if it is above 200 means it is generally solid only. Right? So like this. Next, molecular formula. Next, go back. So IR spectrum also very important, which will actually identify the functional groups. So whatever the IR spectrums that you are having, that IR spectrums, each and every functional group has its own wavelength, and that wavelength you should identify and show that your compound, since it is having this functional group, that wavelength is seen on your IR spectrum. That you have to conclude. Next, mass spectroscopy. So here 460 mass. So 460.0. You can see 461 mass peak is there. So 461 means it confirms that the compound is formed. Next. Now this is the NMR spectrum. NMR spectrum actually it confirms the actual structure, proton NMR. So you to make it very easy, what I have done is you also do like this, right? Aromatic proton, one color. General protons, aliphatic protons, another color. Amide protons, another color. Hydroxyl protons, another color. If you change the color and according color if you give it, you can easily explain. I no need to remember to present, whereas generally whenever you present NMR spectrum, The examiners will ask directly, where is this particular hydrogen in your spectrum? This peak corresponds to what? Already if you add some colors, easily you can present. Aromatic protons are here. Even if I don't remember also I can. So use such type of strategies to escape from critical questions generally asked by the examiners. Next. Yes. So this finishes our presentations, right? Please make sure that whatever the topic that you take, present it, write it as simple as possible, as effective as possible. Even a layman should be able to understand whatever that you write, whatever that you present, and that can be considered as a standard scientific structured protocol, right? Thank you. Thanks, Mananda.